All right, and we are all back. And is Anushka ready?
Yes.
Excellent.
Great. So, hello everyone. I am Anushka, an organizer with the BAM, and welcome to our event today. We have two great talks on commercializing emerging technologies. And so let's get started with that. First, I would like to start with a little bit of an introduction to BAM. So we are a team of microfluidic enthusiasts, mostly situated in the Bay Area. And our goal is to bring together people to foster innovation and collaboration within the field of microfluidics. And with COVID around us, we've been doing that by organizing these events over Zoom for the past few months. So if our team members could introduce themselves briefly, starting with Yatian.
Hello, everyone. My name is Yatian. I'm one of the co-founders for BAM, Bay Area Microfluidics. And so I'm currently a research engineer, and I just recently relocated to the Seattle area. And I have been in the microfluidics field since my graduate school. And it's very exciting to see we have two excellent speakers today. And I hope you will enjoy the event, and also looking forward to meeting you all.
Hi, everyone. I'm Thomas. I'm a PhD candidate at Berkeley in Lydia Sohn's lab. I am, not surprisingly, working on microfluidics.
Hi, guys. I'm Adam. I did some microfluidics during my PhD to poke worms and see how their touch neurons react. And now I'm working at a company developing sensors for natural gas leaks.
Hello, everyone. I'm Samira Darvishi, and I'm the Director of Engineering at Casper Bio. And I've also been toying with microfluidics for over 12 years now. And I'm excited to hear all the talks today, and also this collaboration with MAMNA.
Yeah, so hi again. I'm Anushka. So besides BAM, I'm also a PhD candidate at UC Berkeley, and I'm working with Professor Aaron Streets, and my research involves building microfluidic tools for single cell analyses, and I've been in this field for about 6 years now, I guess, including my undergrad. Okay, so moving on. Yes, so you could reach us at our website, or you could contact us through hello@bayareamicrofluidicsnetwork.org. We also have a LinkedIn group, and we would love to see you there. Join our group. And then, as Samira mentioned, this is a special event because we are collaborating with our friends on the East Coast, MAMNA, the Mid-Atlantic Micro Nano Alliance. And so I would pass on the floor to the organizers from MAMNA to introduce themselves.
Sure. So I'm Heidi Leonard. I'm the MAMNA president. I'm currently the head of R&D at a startup based out of University of Maryland, College Park called PathaTrack. I completed my PhD a couple of years ago doing microfabrication and sensors. And during that time, I met Thomas at the MicroTask conference in Georgia, Savannah. So yes, we're the Mid-Atlantic Micro Nano Alliance. We are bringing researchers together and to network, to look at different capabilities that we have, create innovative ideas, and spark new applications and new opportunities for commercialization of miniaturization technologies. So we're a group of industry, academia, and a lot of governmental institutions, since we're in the Maryland, DC area, Virginia area. And we've also been at least trying to host various events like virtual seminars, workshops, and symposiums for our members to network and share what they've been doing and what they're up to. And so with that, I'd like to introduce to you Ryan, my co-president.
So hi, everyone. So Ryan Sokol, I'm co-president along with Heidi. So I'm an assistant professor at the University of Maryland, College Park. My group focuses, oops. Okay. And so I being muted and unmuted a lot. I'm going to pass it along maybe, because I'm not sure how long I'm going to stay unmuted. But basically, okay, all right, I'm going to pass it on to Oliver, and we'll take it from there. Looks like Oliver is not here. Okay, let's keep going then.
Okay, cool. Right.
Oh, I'm unmuted now. Can you hear me?
Yeah.
Okay, yeah, hi everybody. I'm Mamna's treasurer, Oliver Barham. I also do micro nano stuff and help keep us from getting in trouble with the IRS. And but the main people are Heidi and Ryan, so I'm here as support when I can. Hi everybody. Bye.
Right. So these are the website and ways to contact the Mamna team. And then, so moving along with our event today, we have two great speakers lined up, followed by a discussion which will be led by Dr. Dawn Arnold. And then at around 6 p.m., we would be doing our networking sessions in breakout rooms that we have been trying for the past few events. So if this is your first event, these are a few event guidelines. So participants will remain muted until the 6 p.m. networking session, and you are encouraged to change your name to reflect your first and last name and any affiliation or where you work. If you want to ask questions, and if these questions are directed to the speakers, you can click on the chat button and send a message to everyone, and then moderators will unmute you and invite you to verbally ask the questions directly to the speakers. If you have questions about the networking session, or if you want to talk to a particular person during the networking session, feel free to send a message to Thomas. And if you run into Zoom issues, then you can send a private message to me, Anushka. So asking questions with the Zoom layout, this is where the chat button is. So you can just click on this chat button here and decide if you want to send a message to everyone to ask questions to the speaker. Again, if you have Zoom issues, send a private message to me. And if you want to talk to somebody during the networking session, then send a message to Thomas. So for the job board, we have a few job postings from Illumina that we will hear about at around 5:50 p.m. And then we have one job posting from Interbio. And so for networking session, everybody would be asked to join a breakout room. You're encouraged to turn on your video if you're able to. And then feel free to hang out and chat as long as you like. As I mentioned before, if you want to switch rooms or if you want to talk to a particular person, you can send a private message to Thomas and return to the main room. If you want to get help from within the breakout room, feel free to send a message to any BAM or Mamna organizer that may be part of your breakout room. And so with that, I would like to pass it on to Yatian to introduce our speakers.
Thank you, Anushka. Our first speaker is Dr. Lily Kim. Lily Kim is a partner and a chief scientist at OS Fund and has contributed to the technology commercialization ecosystem for over 15 years. She has advertised Fortune 500 biotech and pharmaceutical companies, helped the research group at the Wyss Institute at Harvard commercialize their work, establish a community forum for microfluidics commercialization in New England, served on the board and directors of the Hertz Foundation, helped neurotechnology company Kernel get its start, and advertised multiple founders and investors. Lily has a bachelor's degree in electrical engineering and a PhD in biomedical engineering at MIT and Harvard, where she was awarded a Hertz Fellowship. So after the bio intro, I also want to share some story about Lily. And so for some of the members who are in microfluidics, you might know we have a very close partner on the East Coast, which is called Fluid Maps. So Dr. Lily Kim was actually the founder of Fluidic Maps. And also in 2017, when Thomas and I first met, we wanted to build a community on the West Coast. And so looking at the example of Fluidic Maps, and then we reached out to Lily and asked for her advice, and also her seeking for her advice, how should we build a community or organization like that. So I had a phone call with Lily, and she gave me like really good advice and support. And also one piece of advice, and she gave me, I still remember, was that just set a deadline and just set the deadline of the event. And so just to start to send out the event and invitations and to let people know this is something we're going to do. So I was really glad we followed her advice. And so now we're building a very strong community on the West Coast. So it's really an honor to have Lily here today. And so we really look forward to your talk.
Thank you so much, Yatian. I'm so excited to talk, and it's such an honor that you guys asked me, and I'm so thrilled to see what you guys have built. I mean, it's such a testament to all the work that you've done. So thank you, and I'm going to share my screen now and get started. Okay, so today I'm going to be talking about commercializing science and technology. And I initially was trained in biomedical engineering. I was doing microfluidic perfusion of embryonic stem cells back in the day. But after I graduated, I actually transitioned to business roles. And so I've spent some time straddling business and biology boundaries. And so that's part of what I'm going to be talking about. Let's see, I'm trying to get to the next slide here. So science and business can feel really different, worlds apart. Who the alien is might depend on your perspective. If you're the scientist, you might feel like the business person's the alien, talking strange language and behaving differently. And if you're the business person, they often feel that way about scientists too, with the technical language that scientists and engineers can use. And they're definitely different. Biology, for example, is studying living organisms, and business is selling things. That's really quite different. But in some ways, you could say there's some similarities because business is about selling things to living organisms. And so a lot of the challenges that you have in biology, because living organisms are complex, you don't know exactly how they're going to behave or what's going to happen to them. You have to have a lot of trial and error sometimes because you don't know, because of the complexity, you don't know what's going to work, is actually also kind of fundamentally true of business as well. Selling things can be really hard in terms of understanding what other people are thinking. And it's really, really hard. It can be really, really hard to change other people's behavior, to get someone to actually buy something. And so you might think back, when was the last time you sold something? For some of you, it might be, last week. And some of you, it might be, well, maybe Girl Scout cookies in elementary school or something, but you can think, how did it feel to do that? What was that process like? And so, living things are complex, unpredictable. And so, when you're talking about a biological system, there's all these factors you don't know about. Will this plant grow or die? Does it have the right nutrients in the soil, enough light? Is it infected with something? What kind of plant is it? What are the genetics? In the situation on the right with the people in the business room, you've got these people. The woman in the middle is speaking. Maybe she's trying to accomplish something you don't even know if the other people are actually listening or not. And then even the ones that are listening, they might be hearing different things, or they might be thinking about lunch or something totally different. So it can be really challenging. Commercializing science can be really challenging because in addition to getting the science working, to get a working product, you need all these other things, and they're not nice to haves. They're actually necessary. It's sort of all of the above to get not only a working product, but also customers, and also a scheme that could be profitable someday, something that is manufacturable at a reasonable cost, something that you could defend. And every situation is going to be different. For some problems, it might be really easy to manufacture, but it's hard to get customers. Or for other problems, you might have a situation where manufacturing has been the bottleneck and some of the other pieces are there. Or you might have a situation where a lot of them are challenging. And so the tools that you have when you're in a company to deal with these, try to get all of these things happening at once, are your team, the knowledge of your team, the connections, which are really important, the equipment you have access to, and the funding. And so this is why
this is a really highly constrained problem. You're trying to have all these things true at the same time. And so this is part of the reason why startups end up pivoting so much, because they're trying to maneuver their way and figure out how can they get in that sweet spot where they have those things true at the same time. So automation is something that has been applied to both biology and business in terms of looking at computational methods and microfluidics platforms to do high throughput screening. On the business side, sometimes you have some kind of databases or computational platforms that might help you prospect sales, but a lot of business work is still really manual and, basically analogous to pipetting. You have to call people up, you have to talk to them, and you may have to, just like you may have to do an experiment, hundreds of times to, well, hopefully not hundreds, but it could be hundreds of times to get it to work. You may have to do business calls hundreds of times. And there are a lot of different ways that things can go wrong. Your technology might be really powerful, but you might choose the wrong first market to apply it to. And, it takes money to do everything and then maybe run out of money. You might not have identified a first market because you've been working very hard on the science, which is important, but need to work also in the business case in order to really develop the whole concept, the integrated concept of the company moving forward. Sometimes companies struggle because they don't realize that there's certain team skills or tools they need to accomplish these things. Sometimes there's an underestimation of the potential slow adoption in life sciences. So just for fun, I put this science article from 2003, like when I was in grad school, talking about how they're like, oh, like, okay, I've just covered up the just covered up the thing for myself. Okay, life sciences have taken only slowly to microfluidic methods such as lab on a chip, but recent advances may make the technology more compelling. So that's back in 2003. You didn't have any. You barely really had any microfluidics companies. You had some, but there wasn't any cell therapy back then. There wasn't single cell genomics. It was totally different world. And of course, maybe your technology doesn't work and you could be outrun by the competition. And there are many others. But success definitely does happen. I think that it's a really exciting time for microfluidics right now. There's all these companies that are doing amazing things, and it seems like the industry is growing and maturing. So it's super exciting to see. So what I do now as an investor is try to understand, what are the factors that can lead a company to be successful versus not successful. But what I found is studying these companies is really pretty hard because sometimes it's hard to get good information. A lot of the information is private. Most of the public announcements about the companies are really just press releases about they raised this amount of money, but it doesn't really tell you what the company's doing or how well it is doing. So you can kind of talk to people, sometimes off the record to learn, but a lot of this information is still very anecdotal. Just because one company says they did it this way or they had this experience doesn't necessarily mean that same advice will apply to your situation. But I think it's still really important and helpful to get a sense for the range of types of things that can happen out there. So now, given that, I'm going to quickly tell a story, an anecdote of a single company, and I guess you can decide for yourself what to take away from it. This is not a company that I was part of, so I'm really telling the story based on information that's publicly available. But I did meet at one point some of the people at this company. So in 2001, in Boston, there was a company founded called Living Microsystems. It was founded, there were some affiliations with MIT and Harvard, Mehmet Toner's group at Mass General Hospital, and this their website at the time. So I pulled this from the Internet Archive's Wayback Machine. So that's why this picture looks terrible, because that's what the Internet Archive had. But it's kind of cool to see. They wrote, "Our mission is to utilize living cells on chips to produce major breakthroughs in diagnostics, therapeutics, and drug discovery." So that's really broad as a mission. And you kind of get the sense that they had some kind of exciting technology regarding living cells and chips, but maybe they weren't exactly sure what they wanted to do with it yet, but they wanted to do something, try to make an impact, do something important with it. Okay, you go to 2002, 2003, and their website has changed, it's become much more specific. And now they're saying that their company objective is to have to make non-invasive prenatal screening and diagnostic tests become the standard of care and replace amniocentesis. So not sure how often amniocentesis is done these days, but that was 2001, 2002, 20 years ago. So a long time ago. Basically, if you were pregnant and you were trying to find out if your baby had any chromosomal abnormalities, what they had to do was they had to take this giant needle and like jab it into your uterus and pull out some amniotic fluid and try to capture some fetal cells and analyze them and see, look if there were chromosomal abnormalities. And it was generally regarded as a generally safe procedure, but like everything, nothing is 100%. And so there were risks of miscarriage. There, were some small risks of infection. So you can imagine that if you're a pregnant woman, it'd probably be nice to be able to find out this information without having a giant needle plunged into your uterus. So this is what the company decided it would do. And they were able to raise money for this. This is now 2007. This is, 6-7 years later. Here they're pulling in $20 million. They had, I believe, raised some money earlier. And over the course of the company, I believe they raised at least about $87 million. But from here on, it starts getting a little interesting and different. So here, in 2008, they published a paper on what they were doing, and they have this microfluidic approach to isolate fetal red blood cells, so the fetal blood cells are nucleated from the peripheral blood of pregnant women. And you don't see it here, but in the author information, it actually shows that the company changed names and actually moved to Menlo Park from Boston. So they changed coasts. I think some of the scientific team was still in Boston, but the company changed coasts and changed names to Artemis Health, but they still sound like they're basically doing the same thing with probably similar technology as what they started with. And the concept is basically to start by doing size-based separation of the cells. So they've got the fetal cells are much smaller than the adult red blood cells. And so at first they do a size-based separation where the fetal cells are small enough that they just get carried along in the fluid stream, but the adult cells are so big that they basically bump into these posts and get deflected away to the side. So it's a little bit hard to see depending on, I'm gonna move my Zoom configuration here at the bottom. It's not only the fetal cells, which are the NRBCs, but you also get some other types of cells, white blood cells as well. So they did a second stage of cell separation based on the fact that the fetal red blood cells had some sort of paramagnetic properties and the white blood cells didn't, so they were able to do magnetic separation. Anyhow, that's the engineering story there. But then if you look 1 year later, then you see that the same company, Artemis Health, licenses prenatal cell-free DNA analysis technology from Steve Quake at Stanford University. And this doesn't show you the whole press release, but if you read it, they actually are saying, okay, we've licensed this and it's a great complement. We've got cell-free now and it's a great complement to our existing cell-based test. And that's it, basically, yay. Okay, great. Then, okay, 4 years later in 2013, they changed their name again to Veronata Health now, and they're sold to Illumina for $350 million. Great outcome, amazingly, but it's purely for the cell-free DNA test that they acquired the rights to develop that IP in 2009. So what really fascinated me about this story was just a lot of times when you hear about startups, you don't always hear about all the different twists and turns they go through. This one changed names 3 times and some changed coasts and changed some people and changed their fundamental technology. They had been working on this one technology for 8 years, basically, when they changed. And then the last 4 years, they changed to a completely different, they totally went with a completely different technology, which ended up succeeding. It's not totally the end of the story yet, because when I was researching this, I came across this paper, and now, like in 2019, there was a group from Australia that was saying, you know, maybe we should look again at circulating fetal cells for non-invasive prenatal testing, because the cell-free stuff that's now gained broad clinical acceptance can't give enough information about the full spectrum of chromosomal abnormalities. So they're saying, we think that circulating fetal cells might come back to the clinic in the future. So this was a fascinating story to me. It sounds like it was very technologically challenging to do that initial development. And I didn't say, but in that paper, they say that they were able to improve on the existing methods 10x to 20x on what people could have done previously. But I guess perhaps it just wasn't good enough at the time. So that's the talk. And basically, I wanted to talk about, you know, how business in some ways can be seen as similar to biology. There's a lot of blood, sweat, and tears that go into it. And so it's really important to start the business thinking and concepts early and in an integrated manner with the technology development. So thank you.
Thanks, Lily.
Great, thank you, Lily. And let's see if we have any questions from the audience. People may be typing in their questions.
Okay. Let me, would you like me to stop sharing my screen, or?
That is fine. Oh, I see we have one question come. And we can unmute. This is a question from, let's see.
If you want to ask yourself, Evan, you can go ahead.
Oh, I can see now.
Oh, am I unmuted?
Yeah.
Yeah, I was just wondering, fascinating story. Did any of the original founders from 2001 benefit from the final sale to Illumina? I mean,
that is a great question, and to be honest, I don't know the answers to that on what the numbers look like on that. I actually met this, I forgot to say in the talk, I actually, when I was in grad school, I visited this company in Boston because I was trying to learn what I could do after I got a PhD in microfluidics at the time. And I met them, and I actually contacted a few of them last week, but they were a bit busy, so they haven't got back to me yet. But
yeah. All right, I don't see any other question that's coming. And thanks again, Lily, for your talk and also fascinating story. And so if people have other questions, and so we will have a panel discussion later, and we can definitely have more deep discussion during the discussion. All right, so now I'm going to introduce our second speaker, Dr. Elizabeth Mirowski. And Elizabeth, or Ella Mirowski, started as a SBIR/STTR program director in May 2020. Before NSF, Ella was a founder and CEO of Vibralium, a medical device startup. At Vibralium, she built a strong collaborative partnership across industry, academia, and federal labs to develop a novel solution for clinical imaging that resulted in new products and new market segments. She also worked for High Precision Devices, where she successfully transferred several technologies into prototypes and commercialized them, creating a standalone revenue-generating company, Qualibre MD. I'm sorry if I mispronounce any of these companies. So in just 3 years, as a principal investigator on SBIR grants from various federal agencies, Ella led the program direction, including research, engineering, manufacturing, and market development. In addition to these activities, she engaged in fundraising from venture capital and private equity sources. For more than 17 years, Ella worked and consulted to small business, managing the development of technologies involving photonics for display, semiconductor electronics, renewable energies, nanomaterials, and the lab-on-chip platform for evaluating neural growth and biological process and the single molecule level. Ella holds a PhD in physical chemistry from the University of Colorado, a bachelor degree in chemistry from Columbia University, and a completed National Research Council postdoc at the National Institute of Standards and Technology. And very impressive background. And now let's welcome Dr. Mirowski.
Thank you for that introduction. I'm going to share my screen and we'll see if it works here because I had a little bit of trouble earlier. And then also I live in a, how do I say this? My internet comes from Wi-Fi off of a mountain across a valley and it's mounted to a tree. So with that said, I'm going to turn my video off so I have enough bandwidth and there isn't too much issues with this, but let me just go up. So many screens in this day and age. So I'll stop the video and I will start this presentation. Hold on one sec. All right. I am trying to Sorry.
It looks good.
I know, except I'm trying not to have it all completely go away. So we'll do it this way though. It'll be fine. All right. Well, great. Well, thank you for all attending this. It's a great turnout with 56 people. So I'm looking forward to getting a lot of questions from you, hopefully, because we have a great program. Basically, we're looking at taking inventions and making impact out of them. I'm representing the Industrial Innovation and Partnerships Division at the National Science Foundation. And so basically our goal at NSF is to enhance our nation's security, drive the US economy, and advance our knowledge to sustain global leadership in innovation. As a whole, NSF evaluates 40,000 proposals each year to determine where their $8.3 billion budget would be most impactful. Of the 11,300 projects funded, 94% of them are for basic research, education, and related activities. NSF supports over 1,800 institutions in the country, 306,000 people, and has funded 242 Nobel Prize winners. About 3% of that budget, of the total NSF budget, is dedicated to seeding public-private partnerships. NSF supports discoveries in 9 scientific areas. The Industrial Innovation and Partnerships Division is under the Engineering Directorate, and our division director is Andrea Bells. As a percentage of total dollars spent on basic research, NSF makes a very significant contribution to many of the scientific disciplines, from physical sciences all the way down to computer science. And what this is basically saying is that 27% of all science and engineering research is funded by NSF. 83% of, say, computer science basic research is funded by NSF. So we make a very large impact with our taxpayer dollars. So because of our comprehensive commitment to empower discoveries in all areas of science and engineering, NSF enables America's best minds to realize their dreams and make a positive impact on society. Over the years, NSF's empowered discoveries have fostered long-term economic growth, addressed critical national needs, and returned enormous dividends to the American people. We've all been able to see the effect of 3D printing and what that has done to revolutionize manufacturing in the United States. But there are lesser known stories that include measuring gravitational waves from space, which enabled extensive collaborations, including those between academia and industry, and really drove a lot of innovation at small businesses in terms of what their capabilities were. Of course, we are always looking to the horizon to find new breakthrough science. And again, NSF budgets about $8 billion a year for exploratory basic research. 272 million is in translational research, which is distributed through the Division of Industrial Innovation and Partnerships. No other division does any kind of industrial academic funding. And basically the goal of the work is to increase knowledge in areas that impact our industries while making a broader impact on society. So the Industrial Innovation and Partnerships Division, which is also known as IIP, so I'm going to refer to it that way since we love acronyms at NSF. We have 5 programs and supplements through which we can distribute funds. The first one is America's Seed Fund, which is the Small Business Innovation Research, Small Business Tech Transfer Research Program. There are many other government entities that have similar programs. Ours is different in many ways in that we basically are looking at the cutting edge science. We don't really have a program where we try to invite people to solve specific problems. We want people to come to us with awesome technologies and figure out where that technology can serve a need in the marketplace, or basically fix a pain point that we have in the marketplace. The other modes of funding are the Innovation Corps, also known as I-Corps. There's Partnerships for Innovation, the Industry University Cooperative Research Centers, and GOALI and Intern. And I will talk a little bit more about each of these shortly. So our focus is on funding deep technology development, but we're also interested in deep societal impact. And here's an example of why it matters. So Blue River Technology developed a deep learning-based see and spray technology for identifying individual plants and selectively spraying them with herbicide, basically looking for weeds and only using herbicide on the weeds, and also for applying fertilizer and fungicides to crops and plants. This basically gave farmers knowledge of what weeds are present and what treatment to use, and essentially reduced herbicidal use by 90%. This company was bought by John Deere during their phase 2 for $305 million. And all of the funding for the startup came from NSF. Another example, because we really have a broad range of technologies that we fund, is Ginkgo Bioworks. And this company engineers organisms for a wide range of uses, including medicinal bacteria, fermented products for new flavors and fragrances. And the process involves designing new organisms based on their code base of cells, enzymes, and genetic programs. These processes that they've developed are more renewable than synthesis from petroleum. They're more stable and efficient than extraction from botanical sources. This company just finished a phase 2 and 2B. They've received additional funding from the Department of Defense and Human and Health Services, which typically NIH, to develop alternate applications of the technology. And their most recent valuation came in at over $1 billion. So how does NSF help researchers catalyze commercialization? Well, NSF funded programs in IIP fulfill 4 critical areas as researchers work to translate their technologies out of the lab. The first is to educate. We have programs to teach them entrepreneurship and how to work with industry. Then the second is accelerating. We teach them how to do customer discovery and really figure out where is their technology going to fit into the market. Where do people want the technology to be? The third is to demonstrate, basically let them build a prototype or proof of concept through our phase 1. And the last one is to translate, which is phase 2, which is commercialization of the product. So America's Seed Fund, which is our small business innovation research or technology transfer arm of NSF, basically has 2 methods. One is a company can apply to the program as itself with an innovation that they want to transfer. Another is that a small company partners with an academic institution and decides to use that academic venue to do proof of concept and feasibility studies. So we just have different ways that the money is distributed in those 2 programs. The SBIR program, the company can only part with 30% of the funds, while in an STTR, the company can part with up to 70% of those funds, so they can really use outside resources if they don't have internal capabilities. So I am currently a program director for the advanced manufacturing portfolio, but I also help with the biological, health, and environmental technology portfolios as well. So we award $200 million per year in funding with up to $1.75 million per company. And we basically fund up to 300 proposals each year with those funds. And the idea is, again, to develop high impact technologies. The initial feasibility phase is 6 to 12 months and a maximum of $250,000. It's actually $256,000. Phase 2 is for completion of the prototype development and starting to commercialize. And this part of the project takes up to 24 months, and the award is $1 million. We also offer a phase 2B, which is a 1 to 2 NSF to third-party investment match of up to $500,000. So this is specifically to allow the company to really try to commercialize this product into the market. So in order to apply to the program, the company has to submit a pitch for evaluation. When invited, they can submit a full proposal, which will be reviewed by experts in the field in 3 main criteria. The first is intellectual merit. Is the idea innovative or a new approach that will solve a big ongoing problem? Is it high risk, high reward? Does it require R&D to prove out the idea? This is typically where angel groups, private equity, and VC are not interested because the risk is too high. So I like to think of NSF as kind of like your friends and family funding to really prove out something that seems really cool, but just is so sort of dangerous to touch for different other parties. Also, we look at broader impacts. So what's the potential of this technology to benefit society and contribute to the achievement of specific societal outcomes? And the third, which is the only criteria that we actually use in the rest of NSF never uses commercial potential as a criteria, but we do. And that of course deals with the market, customers, the value proposition, the team's ability to move the company in this direction, and does it have a competitive business model? So we review on that more on phase 2, because what we have during phase 1 is something called the boot camp, which is a mini version of the I-Corps program. And that really is geared towards helping the team that's usually very technical understand how to do customer discovery, how to do market analysis, and go towards that direction of commercial potential. So then here's a breakdown of the different topic areas we fund and what percentage of the funds went into that area. These areas represent 24 portfolios that we currently have. And our portfolio topics are meant to be a loose guide as a goal just to attract the most innovative ideas and approaches. We don't really need to, they're pretty broad categories. And so we like to keep them that way, because that way we can encourage people to have very different and very out of the box kind of ideas that can come to us. And of course, we've tried in the past to not have any sort of guidance, but that also kind of didn't work and people didn't really know what to submit. So we have kept it to 24 portfolios at this point in time. And as you can see, currently our most popular sectors are life sciences and information technologies. So to give you a little bit of an idea of what a phase 1 awardee looks like, of what our avatar of a company looks like, it's basically 91% of them never had an SBIR or STTR phase 2 award from any agency. 85% of them have 5 or fewer employees. 72% were founded in the past 3 years. And more than 3/4 of the companies are first-time SBIR STTR winners. As of 2014, our portfolio companies have received $9 billion in follow-on institutional financing, and there were 107 successful exits. Moving on to our other programs, I-Corps was developed to spur translation of fundamental research to the marketplace, specifically through training NSF-funded faculty, students, and other researchers in innovation and entrepreneurship. Currently, there are 9 nodes throughout the country with 27 universities and 99 university sites represented. 8 other federal agencies have also picked up teaching I-Corps. And then since 2013, 1,600 teams have been trained and nearly 800 startups were created as a result of the I-Corps program. It is basically a entrepreneurship training program, covers everything outside of the technical aspects of the company and how to be able to deal with those. And I always like to say that people under this either figure out that they have the entrepreneurial bug, or they come with the skill set to be able to find a team and continue to build out their team with people that they know would be good at doing things that you need to do to commercialize a company. The impact of I-Core is in getting the technology into a form that the market and the customers need, which requires going out of your comfort zone to find the information. And it's basically a different type of research than most scientists and engineers are used to. So I-Core is not about selling, pitching, raising money, writing grants or business plans, at least directly. What it is basically it's $50,000 that are given to teams to perform customer discovery, identifying opportunities through relentless firsthand analysis of customer pain points, and using that to figure out if there's a potentially viable business model that you can build your innovation around. It's possibly one of the most difficult things that that people can do, because we ask them literally to go and interview people without telling them anything about what the technology is that they're doing. They are not allowed to bias the person that they have in front of them. So you can imagine how difficult it can be to start a conversation with somebody. So another program that we have is Partnerships for Innovation. And in this case, it's coupled to I-Core in that the faculty and the students can tap into the I-Core program. It also funds $550,000 to translate technology through collaborations with industry. It gets faculty and students engaged in technology translation and entrepreneurship while promoting a sustainable academia-based innovation ecosystem. It's geared towards technologies that need additional basic research to prove out the scientific capability. It basically precedes the market proof of concept and feasibility studies that we fund in the SBIR program. And then well, it's not the last one, but we have the Industrial University Cooperative Research Centers. And what these are is basically NSF provides catalytic funding to an academic research institution that collaborates with other academic research institutions to build a research center. Typically, this money is about $150,000 per year for 5 years. Then these academic institutions find industry members that pay a membership fee to be able to access the combined resources of all of this, the infrastructure of the university, the research labs, the graduate students, the postdocs, the faculty. And the idea is that they perform the basic research that's mutually beneficial to an industry as a whole. So it's exciting because we've had competitors basically become members of similar sites. So you could have a DuPont next to a Monsanto looking to, do some basic research that they know that neither one of them could really undertake themselves individually. So the NSF funds effectively reduce the overhead. So for example, in a 4-university IUCRC, in phase 1, the leverage for a typical member paying $50,000 in an annual membership fee is 23 to 1. What that means is that for every dollar that the member puts into the program, they get an additional $23 of infrastructure and capabilities back to them. So it's a really it's a fantastic program to be able to do some large scale basic research. Currently, there are 804 unique members, 73 centers, 203 research sites in 37 states, and over 1,600 of the center trained students were hired by members. So it's a pathway to a decent job. And then our last program is Intern. And in this case, the Intern supplements fund non-academic research internships for graduate students in an amount of $55,000 for up to 6 months of an internship. A variety of non-academic organizations qualify, including industry labs, startups, government agencies, policy think tanks, and nonprofits. So you can imagine that if you're a startup and you could use an intern, this would be a good way for you to get some supplemental funding to be able to pay for some extra effort on your project. And there are also funds for faculty co-mentoring. International students can participate in the program, and then IP under the NSF trainee policies belongs to the company. So again, we're seeing the same slide. Many people who are interested in translating technology often talk about the valley of death, or the funding gap between basic research and the market. And so IIP funding provides researchers, entrepreneurs, startups, and small businesses with the funding to take on these risks and commercialize their technology. And again, we do it through this four-pronged approach, where first we educate using I-Core, PFI, and Intern as our modes to do that. Then we help accelerate the tech transfer by teaching how to identify the target market and the customer using the I-Core program. Then we help fund the technology demonstration through IUCRC, PFI, and the SBIR STTR program, and then translate that into final product to the marketplace through SBIR phase 2 and the supplements. And with that, this concludes my overview of the NSF programs. We are always looking to support new ideas. And for those of you in the private funding world, I'm sure you see many technologies come across your desk that are just too early to invest in. These are the companies we're looking to help. And so feel free to reach out to me with any ideas, questions, or comments that you have on that. And thanks for your attention.
Excellent. Thank you, Ella. Thank you for the comprehensive overview about the NSF programs. So we're a little bit running behind on time, and so we're going to jump into the panel discussion. So if you have any questions, please feel free to type in, and so we will get those answered during the panel discussion. Okay, so please allow me to introduce our moderator for panel discussion, Dr. Don Arnold. Dr. Don Arnold received his bachelor degree in chemistry from Cornell University and his PhD from UC Berkeley, studying chemical reaction dynamics and molecular spectroscopy. From 1997 to 2000, Don worked as a senior technical staff position at Sandia National Labs, where he changed his focus to the development of microfluidics and microscale chemical analysis systems. In May 2000, Don co-founded a spin-out company based on this technology, Exigent Technologies, acquired by AB Synex in February 2010. In August 2012, he became VP of R&D and principal scientist in AB Synex. Don transitioned to business development in 2015, identifying technologies showing potential for the Synex pipeline. In 2017, Don founded Veristat, where he provides technical and business consulting for life science companies as they navigated from startup to exit. All right, let's welcome Don.
Yatian, thank you. And welcome to everyone who came for the session, and thanks to MEMNA for working with the Bay Area Microfluidics Network to put together and tie the two coasts together. Zoom's made that a great possibility, so it's glad to have everybody here. I'd say it's great talks today by both Ella and Lily. As Yatian said, I'm kind of working with startups, almost similar phases, and seeing tech around that area. And so it was interesting to listen to both of you talk about the approaches and the challenges that teams have coming in. And I know that we have within I'm looking down the participant list, people from a variety of ranges of phases of companies, from startups to large companies that buy startups to venture capital people. So it's I'd say that, one of the things I want to kind of retouch on just to get started a bit, in the space where you have emerging technologies, a lot like microfluidics, things that we see that people talk about at this networking event regularly, you end up having teams that come together who have to be somewhat multidisciplinary just to get the technology work, to start thinking about the business, and then to move that over into a business. They have to bring in even more disciplines unless somebody has been spending some time thinking about that aspect. I know both of you kind of touched on this. I saw differences in the way that you would approach the assistance. Can you comment a little bit on what are the biggest, when people come in to the different programs? It sounds like LSE super early stage folks. Lily's going to probably get a little bit later, but also come in that phase. What do you see as really the biggest challenge that the teams have to overcome to really get to that phase where you feel comfortable providing the funding into the team to get the technology moving?
I guess I'll start since I'm a little bit before. I'm looking
Okay.
at, the people who are like, I have an idea, and a couple other people think my idea is good, and they have some, gumption to jump ship and work with me. So, I mean, really, our program, we've got pretty strict guidelines on what we look at, and so the intellectual merit, oftentimes in phase one, and this was a question in the chat too, is really. That is one location we look at very heavily, right? And then we look at societal impacts. And so you have to really hit those two marks. We even have during the pitch process, we kind of look and see, are the people moving in that direction? So if you give me an idea, it's really broad, I'm probably going to go back to you and say, hey, can you tell me a little bit more about the R&D in the pitch process? What is the R&D that's going on here? Then when I get the R&D, then I'll say, well, is this engineering design, right? Is it just doing some small incremental adjustments to what is already out there? And then some seem like that, but then you realize, hey, we can improve, the resolution of a 3D printer by 1,000-fold. And we can reduce the time by, 1,000-fold. When you start to see these improvement numbers be large, and then you have to dig deep, because then we invite panelists, reviewers, experts in the field that know what it is that might be possible, right? And so you have to have a really good sort of scientific backbone in phase one that says, even if you didn't do the feasibility study, the science and the thought process behind it is strong. And then phase two, we start moving on, and we're like, okay, you got to think commercially. You've proven your feasibility. We believe you met your milestones. You can meet the next milestones in phase two, but it should be going on the trajectory of commercialization. And we completely focus on that, not in phase two.
And as a part of the, quick before you go to Lily, as a part of the I-Core program, I guess, or where you have the boot camp, as they move through that, is part of the program to help find business knowledge expertise for people if it's really not inherent in the team that they have existing already, or are they kind of on their own to go find that through the NSF programs?
So, as with a program director and with the mentors that you have in the boot camp, you'll get feedback as to what you should be doing. But ultimately it's the decision. We don't ever tell a company what to do. We make recommendations, and then it's basically, it's up to the company to go and find.
You eventually find volunteer mentors coming in to help, and they can guide them in a sense, right? Okay,
exactly.
Sorry. Yeah. So Lily, sorry. Back to the question about what is the biggest challenge you typically find coming in with the team?
Yeah, I mean, I think part of the biggest challenge is sort of this science, technology, business kind of, I don't know if I call it divide, but being able to bridge those worlds. The teams that come to us, sometimes they have a business person. They might be a team of, say, a business person, a scientist or a tech engineer who have already met before they come to us. But there are definitely many teams that are perhaps like 2 engineers or a couple of scientists, and so sometimes those then fall into 2 buckets. Some of those people don't want to hire a business person. Other people say, we're the CTOs, CSOs, we're going to find a CEO later, or we're going to, so there's different mentalities, and at the end of the day, you need some people in the business who have business skills. And whether it's people who have a scientific and engineering background, building those themselves, or hiring help. I think even if you hire someone to help you, which can be great, you can learn from them. The sort of original founders of the company, if they're coming from a scientific background, it's really important for them to gain some of that business thinking as well.
Yeah, I think it's a really good, fair assessment there.
I'm a big fan of the I-Core program, and that whole yeah, that whole approach.
Yeah, I help mentor with those the programs out here in Livermore as well. So it's I huge kudos on watching the programs put together for that. With the other thing is quite different, I'd say, between the two programs is how people look at equity. I would think that if you have somebody coming in, I think probably Lily would say that we love the I-Corps because it's a non-dilutive funding in a sense, and we can get things further and then start worrying about equity. Is that kind of how you look at this? If I'm an inventor thinking about the commercialization of a tech and they were to bring it to you and you look at these types of programs?
Oh, you mean how do we look at non-dilutive funding?
Yeah.
Oh, it's awesome.
Yeah,
Great,
that's right.
it's fantastic. Actually, I was excited that you mentioned Ginkgo because we funded Ginkgo. We were in the first institutional round, and actually, it's great to see companies can do really significant things on some of the NSF and other types of non-dilutive funding.
Yeah. Let's see. I think it looks like Ella's answering a couple of the questions on the fly. So we'll take care of that. And then in terms of, well, let me ask Ella before we move into some of those. One question about, one of the key things that I know when you're looking at it from the private sector is intellectual property is a really important thing for getting a tech out and have your company be successful. How do you guys look at that when talking to people who are looking for funding? What's your stance on intellectual property as a means, as a part of the commercialization of emerging technologies?
It's absolutely essential. I mean, the whole, we have to protect our investment in the company and that we don't just send them out to be completely cannibalized. So yeah. It's absolutely essential. There's money for it in the phase 2. Okay. In the phase 1, there's if people really want to file IP in the phase 1, there's the profit that you can take off the top, that the 7% fee that lets you spend it on anything you want practically. So that's one place where you could use it, but typically phase 2 is where we really encourage it.
Yeah. And does, at the end of the day, with you have companies that go out and they get $9 billion total of follow-on funding. I mean, what does NSF get out of that? Just the continued tax funding comes back through into the
that's
NSF
the
budget?
hope that it is about, I mean, it's economic growth. They hire people, those people pay taxes, and and then we all just go and funnel back into the system and send it out again.
Yeah, okay, cool. I can't see if you did this one or not. There's one from Evan. I saw that I heard that companies can only receive one SBIR grant from the NSF. Is that true, or are there restrictions on multiple grants?
That's not necessarily true. That it's possible to fund 2 if the ideas are really that different and that innovative. But ultimately, we are looking to seed individual companies that can focus their technology on something that is going to be commercializable. So it would almost be difficult to say, now I'm going to fund this thing over here. So I'm going, so you're going to divert your attention. So it's not common, but it happens on occasion, but it's not common.
Okay. I'm looking to see here. And then let's see, we got a question from Brendan. Let's see, what's the biggest misconception graduating scientists have about commercializing a technology? Oh boy.
If you build it, they will come.
Yeah.
And a lot of them don't know, like the technology is super exciting. They see what goes on and their struggle is finding what to focus on, right? It's like if you have, the hardest part is having a technology that can really be applied broadly. That is a really hard problem for scientists because they want to do it all and get them all done.
Yep. And so Lily, I mean, do you see a lot of grad students showing up with coming into OS Fund, or you get people coming in at different stages?
Occasionally, I think so. I'm trying to recall. I don't. Yeah. I think there's often people who've just finished their PhD who will come.
Yeah,
and
I
especially nowadays. Yeah.
Yeah, for people, I think talking to people with ideas, trying to get things going, and there's quite often the how do you even get for NSF we've got the link and there's the website. What's that? How does if somebody's got an idea and they want to talk to someone about funding that's within the VC community, then what's the advice you have for them for how to how to get in touch with someone, and then how to make sure that they listen beyond the 30-second pitch there for 30 seconds of the first conversation.
That's a good question. Warm introductions are always a good thing if you know someone who knows someone at a VC firm. But we actually at ours we do also take I think we take submissions to our website. So we have at osfund.co, we have a place on our website where you can, if you have an idea, you can contact us, and we do review those. So other ways, there are sometimes conferences that you can go to, like these. I can't remember what they stand for, but it's like, for example, the Resi conferences are, I think it's early stage science and technology, and you could give a talk there.
Yeah, it seems like events like this, tuning in, like the ones you started in Boston with the fluidic MEMS, if you're in that space, I've seen more and more of the funding groups showing up. You just have to figure out which ones they are. They don't necessarily wave a flag for you. Find someone who knows someone. It sounds like a great advice there.
Yeah, and I would actually also add, especially now during the pandemic, during COVID, especially if you live in an area where there aren't very many investors, right now, everybody's kind of equal because everybody is virtual. So I would encourage people to reach out to people, even if they're from, maybe they're not in the Bay Area, but, they want to talk to an investor there. I would say just go for it and contact people.
Yeah. Let's see, I had a question, but I'm going to go to one of the ones here. So Sixing Lee has the question, what's the approval rate of the SBIR, STTR applications currently, and what are the critical review criteria to go from phase 1 to phase 2? That's one for Ella. Another question about how much you can say about particular programs, but
so
generally,
from the math on one of the slides that I gave, there's 40,000 proposals coming in, about 11,000 get funded.
Okay.
So that gives you an idea. It's about 20%.
Yeah.
And then, like I said, phase 1 is a lot about the technology, the broader impact. We do review on commercial potential. You have to kind of know your commercial market. It's difficult when you throw in a proposal and you don't talk about any of your competitors. Then we have reviewers come on and they're like, there's all these competitors, this technology is not novel. So you kind of, at least in the phase 1, you really should know your landscape and how you fit into it. You don't need to know what the commercialization pathway is, who your customers are exactly going to be, but you should know how your technology would fit in to the market. And then phase 2, again, the criteria still, there's milestones for the technical part, but really there's a whole commercialization plan that you have to include in that. And that is what we weigh heavily on that in phase 2.
Cool. And that one other, Lily had a great story there about the pivot process within going from benchtop to something that a customer can work with, and I can I won't share my long story, but I can certainly assure you that when we were running through our company, we pivoted at least a couple of times on core technology within the device because of either things that you did not anticipate or things that you realized later in the market, either technical or market-wise, just had to change. And it seems we had a great example from Lily within the NSF program. There's a reasonable time period over there. Do you see significant changes in the way the technology has to operate, and how does the grant shift? I know when we were doing it, we had an ATP grant for a couple $1 million, and we had to make a shift in the middle, and they worked pretty well. How does the NSF deal with that? If I'm a company coming in and I'm funded and I'm like, oh no, this doesn't work.
So
are you out, or do you get to regroup a bit?
You do get to. So it's funny because I just sat in on a phase 2B today, and it turned out that they went through phase 1, phase 2, and now they've got this influx of funding coming, and they need to pivot because they realize that the technology should really go to a different place. And so it's fairly easy, in my opinion. You work with your program director. You say, here is the change in the scope of the technical work that we have to do. This is why we're doing it. And we basically just approve it if we see that it is technically feasible and well laid out plan. So, we understand that you have to pivot sometimes, and so.
Yeah, yep. That's true. And do you see kind of the, with Lily? I mean, that was a great story about the living microsystems. Do you see that pretty consistently within the groups that you end up funding? Is that a common event that you see with the agencies, or do most manage to make it through unscathed?
Oh, you mean the big technical pivot there?
Yeah.
Yeah, I mean, I don't have numbers. I've anecdotally heard it from other companies, friends, founders at other companies, kind of like your story, that it may happen more than people think, because you don't always hear what happens inside.
Yeah, I just, it's one of the things I like to bring up in this, because I think if people are just going into it, anticipating it and being ready for, knowing that it's okay for your stuff not to be working as necessary, but then people are going to say, okay, that's great, because eventually at the end of the day, you end up at least for the ones who make it all the way through, you end up with a better solution, whether it's technical or business. So it's,
kind of that, it brings in that sort of sunk cost fallacy of,
right.
Yeah.
That's right. All right. So let's see. Oliver. Oh, our treasurer. So for Ellen, does the NSF communicate and collaborate with the other government SBIR programs? Yeah, that's a good question. If the technology doesn't fall into your portfolio, then could that fall into kind of an Army or a DARPA program?
So we don't necessarily, I don't know any of the program directors in ours that have the bandwidth to really do
the cross communication.
Close cross communication. I know some of us do from relationships that we have. It's something that we've talked about maybe doing more of actually in the future, with now, with just expanding and doing things a little bit differently. We do work within NSF too. So, sometimes the basic research components, they'll say, hey, we see something that's more along the lines of trajectory of commercialization and basic research. And we collaborate that way. And honestly, for me, sometimes I do, and it depends on the program director and what they know and knowledge. When I see something that fits more into Army or DARPA, I may just comment on it in the pitch. Usually it's in the pitch process that we would funnel and say whether or not this fits the NSF program. And so that would be a place that if at least you applied and I was reviewing it, I might say, hey, this might be better suited. We do have to be careful, because sometimes people then run away and they say, oh, so-and-so said that this fits here, right? So you kind of have to really phrase things appropriately, because we never tell anyone what to do. We may make a suggestion, and it's something that you have to decide that you do, because this is your life, this is your technology, and it only works if you do it kind of the way that you see it done in terms of with guidance. I can't, there's no solution, right? We know all these books that wrote about success stories for businesses, and nobody's been able to reproduce any one of them. So
yeah. Okay. And there's a good follow-on in the question of how would you compare the NSF to, say, the NIH SBIR programs in terms of the process or the priorities and such? Are they, is it micro? I agree. I look at, you can have a microfluidics program going into either one.
So we don't fund clinical trials for pharmaceutical technologies, things like that. That's typically an NIH. But we do look at those fledgling ideas that are applied to life sciences. As you could see, we had 20-some-odd% of the portfolio being delineated there. As far as the evaluation process, so the NIH process has panels that they have put together, and there's consistency in terms of that reviewer pool. However, that reviewer pool is basically, they're the ones that are determining whether or not your proposal gets funded, and there's a moderator, and they basically just moderate. At the NSF, we have a peer review. We can either do an ad hoc review system where we send out proposals, and then the program director gets back, reads the reviews of the proposals, and then makes a determination based off of those reviews. And we also have another process, which is a panel review. So we send out the reviews to the reviewers, they write their reviews, then we invite them to come together, and then there's a discussion that ensues for several hours about each of the proposals. We always try to say, please don't rank these proposals against each other, because that's not what we're looking at. We're not trying to get a ranking of what's better, what's not. We could fund all of them in this panel session. We could fund none of them. So So it's very different in that respect. And then ultimately what happens transpires, you get a panel summary, basically one of the reviewers writes up a panel summary, and then ultimately the program director looks at all of the different technologies and then makes a determination of what to move forward.
Cool. Okay. Let's see. Yeah, and then I had the one thing I recall, we didn't talk much about the IUCRC, but I remember that there's the one down at UC Riverside is a microfluidics one. Abe Lee, he used to be DARPA, I remember, and then had a NASA program down the now it's cadmium, I think. I was just checking, but I think that's a good point there. I don't know if you know much about that one. I thought of it and thought of it as a good reference point as a promo for a program that's one of the big interagency. I participated as a company member, but then changed company, so no longer participating in that at this point, but still know the teams. It's a great program. Seems like it's a nice way to get some non-dilutive funding and leverage there as well. So I think right now, I just want to transition into the next phase, but I just want to make sure to thank you guys again. It was great talking to you guys beforehand. I love the talks. I think this is a really important part of getting technology out. Both groups have the same passion, right? Taking a tech that's going to be really impactful and getting it out and putting it in the hands of people, and both have the challenge of trying to figure out how to get people educated and able funded so they can turn it into products that can be useful in a reasonable amount of time and with a reasonable amount of funding. So hopefully the audience like that. I think I'm going to switch over to, I'm not sure if it's Yatian or Thomas or Anushka. They're going to run up and set up the job board part, and then we'll move into the breakout session. So hang around here.
Hi, so yeah, thanks to all of our speakers. So the next section of our little presentation here is our job board. So we have job postings from 2 companies tonight. The first is Illumina, and I believe that Tessa's on the meeting to speak about these positions.
Hi everyone. My name is Tessa. Just for background, I actually know Yatian. We both worked at Purigen Biosystems together. And then for myself, prior to that, I did work at Bio-Rad in the Digital Biology Center, working on droplet microfluidics. So today I'm going to review, thanks Yatian for putting it in the chat for all the links. I'm a hiring manager. I lead the systems engineering team, or one of them at Illumina. So the engineer 2 role, that is the one I'm hiring for. We are looking for someone with mechanical engineering background, a little bit of programming, and is interested in DNA sequencing. We're kind of the interface between consumables and the instrument itself. The senior mechanical engineering role, that is also within the engineering teams that I am a part of. That one is looking for somewhat of a leadership role in mechanical engineering and design. The engineer 1 fluidic systems one, I think that one is a bit farther along in the process, so it may not be looking at right now for current candidates, but the last 2, scientist 1 and the staff engineer, are coming from teams that are looking for microfluidic engineers and scientists, and kind of have that balance between microfluidics and biology. So please feel free to reach out to me. And I do think Elizabeth Rosas is also on the line. She is as well from Illumina. So please reach out to us and let us know if you have any questions. Thank you.
Thank you, Tessa. So next up, we have Intabio, sorry, Don. And Don will be presenting this job position.
Yeah, so it's one of the companies I'm supporting around the Bay Area is Intabio, and I looked through the list. If anybody's on the meeting tonight, they can stand up. I know they're exceptionally busy right now. They're basically just relocating into a new facility in Fremont. The company is building a platform for doing intact biologics analysis for kind of pharmaceutical biologics-based therapeutics, and it's a microfluidic chip system that integrates in with mass spectrometers to do very important types of analyses in those workplaces. They just launched their product based around that particular chip that we have up on the screen, and you see the version there that's being launched, have partnerships with 4 different mass spectrometer companies and Protein Metrics Bay Area data analysis, excellent data analysis program. So they have a number of positions open. They've been hiring very actively during the COVID session. It hasn't, Zoom has not stopped them from growing the company. They're up over 30, 35 people now. And as they move into the commercialization phase, they're looking for new people, East Coast and West Coast. They need technical sales manager on the West Coast here, a mass spectrometer scientist, preferably understanding how at least electrophoresis works would be great. They'll be based in the new Fremont facility across from Tesla. They have an opening for a system integration scientist. They need 2 field application scientists, one on the East Coast, one on the West Coast. And they'll need a quality technician as they're ramping up the manufacturing there in the new facility. So the details for those positions are on the Intabio website, Intabio standing for Intact Bio. And you can send inquiries to hr@intabio.com, or you can talk to me after in the breakout room if you want. I don't have all the details, but I can give you some idea. And that's it.
Thank you very much, Don. Yep. So last but not least, we have our networking session. So Anushka, can you actually pull the slide up for that? Great, thank you. So if you've never joined one of our meetings before, how this works is that everyone will shortly be asked to join a breakout room. Please turn your video on if you're able to, and you'll be unmuted and stay in chat as long as you like. If you'd like to switch rooms, there are going to be 5 rooms. And if you'd like to switch rooms, just go back to the main room and message me and ask to join a particular room. Or if there's a particular person you want to talk to, it can then connect you. If you have trouble, send a message to the BAM or MAMNA organizer in your room. So with that, I'm going to open the rooms and we'll see you all soon.